Yeah, what's up? The whole PowerPoint. Mhm. Yeah, the PowerPoint is due next week, and the actual presentation will be later on when we get to Unit 4. What even is that? All right, we're doing chapter 14 today. This is epidemiology, and then probably we'll have time after this to run through chapter 25 real quick. Pretty much what I covered, if you haven't watched the review yet or didn't join in on the review, pretty much what I covered in the review is pretty much what I want you to know about chapter 25. So there's not really a whole lot to say about it, but I will try to run through it when we get a chance. Okay. Starting out, epidemiology is just a field of medicine. It's going to be studying disease or any other kind of adverse occurrence experienced by population. So not always going to be infectious disease. I do want to be clear about that. They consider themselves to be investigating the who, what, where, when, and why of these situations. Starting out with a couple of historical examples, we have Florence Nightingale. Florence Nightingale was a nurse during the Crimean War. She realized that 32 out of 100 soldiers were dying in field hospitals, not from like their wounds, not from gunshot, not from stab wounds, but from dysentery. So that is, in case you don't know, due to improper poor hand washing practices. Florence Nightingale, she improved the whole, basically asepsis of the hospital and reduced that down to 2 out of every 100, basically employing hand washing and changing bedding after patients and stuff like that. Then we have John Snow. John Snow, he was a doctor in the mid 1800s. He realized that there were people who were having cholera. He mapped it out around the town and realized it was around this specific water pump. And it did, in fact, turn out to have cholera bacteria in it. And people who had cholera were going to go get water from it and were just reinfecting it and all this. So he told them they needed to stop drinking that water, but they, of course, weren't going to listen. So he just had that handle removed so that people couldn't use that location anymore. So this was how he had mapped it out. We have these darker like spot areas of households or other locations that did in fact have cholera, and it just is going to be centering around the center of the city where the water was coming from. And then of course this picture of death providing water to everybody. If you don't know, cholera is no joke. It's not just a disease that, you know, you get on the Oregon Trail. It's also one of the worst types of diarrhea you can have ever. So it doesn't cause dysentery, but it causes what they call rice water stool. Literally, what's coming out of your rear is going to be looking like rice water, actually. No brown to it at all. Like it is like clear white. That's what it looks like, because your body is expelling so much water and so much electrolytes that you cleared everything out. There's nothing else to push out. And it gets so serious, people will die from dehydration from that. Of course, that's going to be the number one thing. The treatment for it really is just pushing fluids. There's not really any other way to treat it. They have these beds called cholera beds that they'll put you on, and it's like literally just a cot with a hole in it and a bucket underneath. And that's how they have you wait it out until you get better. Cholera? Yeah, wow. Yeah, people don't realize that it is actually one of those fecal oral route just type transmissions. And we don't see it a lot in the US because it's not endemic here. But if you travel to other places like Haiti and places like that, like when there was the earthquake in Haiti, we had a lot of doctors going over there to help out with that. A lot of them wound up with cholera because people were in like really close living situations. Sanitation was poor, as tends to be the case on cruise ships, not because people are nasty so much as like just a few people need to be nasty on a cruise ship because they're touching all the same surfaces as everybody else. You're stuck in a very small space for long periods of time with one another. So it really doesn't take very much for those locations to either have cholera or things like norovirus, which is one we hear about the most there. But anyways, back into these terms, quite a lot of terms for chapter 14. So do be aware of what each of these terms mean. Be also aware of the differences in them so you can pick them out. Prevalence of disease, we're talking about the total number of existing cases for the entire population. Okay, so it's an overall big picture look at things. Whereas incidence, we're talking about the number of new cases over time. So how many new cases might pop up over a month or something. That is also called the morbidity rate. Okay. Whereas the mortality rate is the number of deaths due to the disease. Okay. All right, this is talking about epidemic curve patterns. I don't really think that's really accurate. So let's talk about really more or less what these slides are talking about, which is dealing with how your epidemics will be starting, okay? Point source epidemic. The point source coming from a single point of source of a person as well as a point in time. So everybody being exposed to one person all at once and all getting sick due to that one exposure. So if I were to come in here with a new strain of, Ebola, and I'm bleeding out on everybody in here, and everybody in here gets Ebola from that one exposure, then that is a point source epidemic. I mean, we could go with other less exciting things, but why would we? So common source epidemic, you have a single source where it's all coming from, but maybe it occurs over a period of time. So that is exactly like what we talked about with the water pumps for cholera with John Snow, right? People were going to those water pumps over days, but it was all that water pump that was causing everything. So that's a common source epidemic. It's over time, whereas point source is one source of the infection and one time exposure. And we have propagated epidemics. We've gotten from very specific to a little more broad. Propagated epidemic, these are infectious agents that are communicable and are maintained over time, like what we often see with influenza. It's usually going to hang around even during seasons whenever it's not as common. It's usually somewhere around. So that's a propagated epidemic. All right, the index case is the first patient that's found in the investigation to have the disease. It's not necessarily the source of the disease, just the first person that they found with the disease. Endemic is a term that I have been using since the beginning of the course, and hopefully it makes sense to you. But endemic is an infectious disease that will exhibit a steady frequency over a long time in a particular area. Relative number of cases that you might see of flu during certain periods of time in Oklahoma. That's what we call endemic for that area. Whereas sporadic That's going to be some random cases in random locations and random intervals. So if you get bit by a raccoon tonight while you're taking out your trash, that's going to be sporadic, right? That's not going to be endemic. That doesn't usually happen. We don't see any cases of that. So an epidemic occurs anytime you are outside of what we are considering to be endemic or sporadic. So more cases than what you would consider sporadic, or way more incidents going on with the endemic diseases. So way more people getting sick than what you're used to. There are exact percentages and numbers associated with this, thresholds that you would have to reach according to each disease, according to epidemiologists, but that's the idea. You're deviating outside of what is expected. That's an epidemic, okay? And a pandemic is just going to be an epidemic that crosses continents. It does not have to be all over the world. It just has to cross continents, okay? All right, we have a way to basically look at the progression of an outbreak or disease, which tends to follow actually the symptom curves that we were talking about previously. So you remember what we had talked about was incubation and then prodrome, and then acute, and then of course I ran out of space, so convalescence. So remember, this is going to be no symptoms at all, first symptoms, high symptoms, like the worst of it, and then this is reduction, right? So we talked about that as being the course of basically an infection, how it's presented in people. This way that the epidemiologists like to look at the history of a disease, so a specific outbreak that we're talking about, its own history, kind of follows along with that pretty well. So we have a susceptible host, someone who is not infected, but could be. They're exposed, okay, and they're exposed and they get infected, and now they're in a subclinical stage of disease. That usually is going to mean that we're in the incubation period. So it's subclinical, you are infected. Your body might be undergoing pathological changes, but you don't have any obvious symptoms yet. So once we have the onset of these symptoms, that's going to be the prodrome, the first symptoms that we're seeing. Then we have clinical disease, which is usually where people are going to be going to the doctor. This is usually the acute phase. And then we have recovery, disability, or death. Those are your three options. You literally have to be 1, 2, or 3. Okay, those are your choices. And that would be happening during and after convalescence, which is reduction of your symptoms. So that lines up pretty well with that theory as we look through how disease progresses. Okay, this is just going through everything I literally just described to you. Next, we have screening tests. Okay, these are different, like it says, different from diagnostic tests, because we want to be quick, easy, quick and dirty. We want to be able to get through it as quick as possible. So they're usually screening tests, less invasive, less hazards involved. Patients are usually more willing to go through the screening tests, but they are going to have the lower accuracy than the diagnostic tests. Okay. So diagnostic tests usually what you do after you have a positive on your screening test. So here we go. Usually those screening tests are supposed to be cheap and easy to perform, and we can deploy them. This part, I like this part here, deploy them on healthy populations that are not really super inundated with the disease yet, or even not at all. And have them start testing regularly so we can detect the disease before it starts causing harm to the population, before it's hard to get control of. Those screening tests are effective for that because they're cheap and easy to use. You don't need much training to do them. So positive screening test usually is going to be followed by a more accurate diagnostic test, just like we have with pap smears. They are screening tests. You have just a brush on the cervix, you send it into pathology, and they tell you whether there are abnormal cells or not. If there are abnormal cells, they're not going to be able to tell you much information about that outside of that. So you'll need to have biopsies done to see how deep the abnormal cells are going. Is this actually some sort of cancer, or is it just abnormal growth of the cells and that sort of stuff? But the biopsy is much more invasive than pap smear. Specificity and sensitivity. So these are often confused by people. I'm going to try to walk through it and tell you how I remember it. So specificity. This is determined by how many people we call positive who are actually negative. So specificity, we are going to be avoiding false positives. You don't want to be calling people positive when they don't actually have that disease. They're being more specific about who we're calling positive. Okay. Sensitivity, determined by how many people were positive, but we incorrectly called them negative. Those are false negatives. So here we're trying to avoid negatives. Sensitivity, the way I like to look at it basically is it's so sensitive, we're going to catch every single positive. Even if they are just barely positive, it's so sensitive, it'll be able to pick that up. So if you can remember that the two terms are specificity and sensitivity, and you remember that sensitivity is, oh yeah, we're so sensitive, we'll catch all the positives, we won't miss a single positive, then you can basically compare those just by knowing that one. You have a hard time with it, okay? Okay, causality, looking at the cause of a disease. We mentioned already Koch's postulates that has to do with diagnosing, finding the cause of a disease, the etiological agent, right? So he took the mouse, well, he took swabs of people and then grew it out and separated out the sample, put that what he thought was causing it into the mouse, it got the same disease. They were able to re-isolate that organism from the mouse that they had originally put in. So that was the cause of the disease. That's how he discovered the cause of anthrax. So that's Koch's postulates for discovering the cause of disease. Nowadays, we use molecular techniques more than those like long, taking forever to grow things out and expose animals and all this. Now we just run things through things like PCR. So remember that correlation and causation are not the same thing. If we thought correlation meant causation, then we would have quite a lot more people being put to jail for murder than would deserve to. So what I mean by that is, if you come up on like a dead body and you lean down to check on the person, you get blood on your hands, and the next person walks in here, and it's just you and that person, you've got the blood all over you. And they just say they killed them and don't even, that's just a correlation, right? That's just correlating those things together. That doesn't mean that that's going to prove the cause, right? There's a lot more that we need to do to investigate that situation to see whether that person did or did not do the murder, right? So that's the best example I can think of, because I know that the news is terrible at telling the difference between correlation and causation, especially if it is, just sensational, basically. So no surprise there, I'm sure. So we're going to be trying to investigate correlation and finding out what the actual cause is, and dividing those two or realizing that they are one and the same. So we do randomized control trials. Here we're talking about having trials like drug trials, where you have an experimental group and you have like basically a placebo group or control group, right? And nobody knows, the doctors don't know, it's double blind, doctors don't know who's getting what, and the patients don't know what they're getting, and then we just collect data, and then people on the other side that do know the answer can interpret that data. So that's the idea with that sort of stuff. So there's some studies that epidemiologists will do in order to try to determine causation and eliminate correlation from that. So we now just definitively say what's causing what. So let's start with cross-sectional studies. We have two factors, like when or how you were exposed and the disease that's being caused, okay? Those data are collected at a single point in time. We're not having any reference to which may have occurred first or anything like that, just at a single point in time, getting numbers on exposure and disease, or where they were exposed and what the symptoms are, for example. Okay, so no reference to what occurred first. There's one point in time that we are taking this information from. And you could look at several points as you go through the studies, but each one point, okay? Case control studies. Here we have a group of people that are categorized as to whether they have the condition or not. And then they're going to be asked questions about their exposures. So they'll maybe ask about, okay, how was your week from the people that actually have the disease? What did you do this week, right? And then people who didn't have the disease, what did you do this week, right? And if there's something that stands out specifically relating to people who were sick, then that they could see as being potential to look down that road for a cause, right? So that's the idea. Comparing sick and not sick and trying to see what the sick people have going on when they do compare it. Then we have cohort studies. This is the one that makes the most sense to me. It's like calm in the storm as far as I'm concerned. But the cohort study is just following people who are not sick over time and then monitoring for any information as that person does become sick. So they'll say, hey, you're joining this study. And you'll be like, cool. And they're like, what do I need to do? And they're like, oh, just write down where you go and stuff, what you eat and stuff like that. Oh, okay. And that's what they do. And then you get sick and they can say, oh, when they ate this, it was 2 days before they got these symptoms. And that's what we've been seeing with these other people that we're doing the study on. That's probably related to eating this food. So that's how a cohort study works. Like I said, it just makes sense. You're going from the absolute beginning of the thing, backing it up and watching somebody that doesn't have it get sick and then learning from that circumstance. Okay, surveillance just refers to watching and collecting data from the population during an outbreak. So we're looking at occurrence, mortality, morbidity, transmission, all of that stuff. Collect as much data as you can about the disease, the outbreaks. There are some diseases that are notifiable or reportable, usually call them reportable diseases in the United States. And these are diseases that a lot of them by law have to be reported. It might be local law, but it might be as high as international law. Like when we were talking about COVID in the very beginning, especially in the early days of COVID, the World Health Organization wanted every single case of COVID reported because they're trying to keep track on how this is moving. And they're trying to keep track of morbidity and mortality. And I don't even know if you know this, but if you go to a doctor and you have a sickness, an illness, and they don't report it to an agency like CDC or local, health organizations and stuff like that, then that data doesn't get out at all. There's no data to be gotten out. So there's no anything for, news to report or any of that. So if you hear on the news that, we have this new outbreak of whatever, it had to be reported, right? Otherwise, that's HIPAA violation. Yeah. So it had to be reported according to law and dealing with the health department and all of this sort of stuff. So health department, CDC, World Health Organization, and there are, of course, highly contagious, highly dangerous, or highly unusual diseases that they're the most interested in, like Ebola, or when we had COVID coming out in the beginning. Are they still tracking it? Is it still a reportable disease? Probably. I haven't checked for sure, but they still want to know about new strains that will be coming out and everything like that. Especially because every other day, China trips and says, oh, whoops, we released another deadly disease. Whoops, that wasn't us. So, reporting the disease basically depends on the physician and all, healthcare providers in general, and the government agencies that might be wanting to watch over this stuff. In our case, the CDC. The CDC releases the morbidity and mortality report weekly. So every week they are releasing a large document regarding all of those reportable diseases that they are tracking and what's going on in the world with those at any given time. It's completely public record stuff. So that is usually the type of stuff where news channels are going to get their information from. Okay, moving on to the healthcare associated infections. Infections that you're likely to get if you were to stay in a healthcare facility. I'm not just talking about hospitals here, I'm also talking about nursing homes. The problem with the nursing home statistics is that issues in a nursing home are less likely to get reported. That's sad, but it is true. So, I don't know. I don't know what to say about that. It is what it is. Of course, we're trying to work towards, providing better healthcare for everybody in every situation. But even so, 1 in 31 hospital patients. So, and then 1 in 43 nursing home residents. So nursing homes doing better according to these numbers. Has a HAI, that healthcare associated infection. So where do they come from? These are the 4 most common ones. They're all obvious, okay? Catheter associated urinary tract infections, central line associated bloodstream infections, surgical site infections, and then ventilator associated events. So not just infections associated with the ventilator stuff, but other things outside of that. as well. So none of these, I'm sure, if you've ever worked in a hospital, can be surprising to you. It's hard to control catheter infections just because it's hard to keep them sterile. Okay. So how do we control these infections in healthcare settings, especially in larger hospitals? Medicare and Medicaid programs will stop reimbursements to hospitals, stop their funding towards hospitals if they are not meeting their required levels of those top 3 diseases, right? The catheter related and the central line and the surgery illnesses. So if you're doing pretty bad according to Medicare and Medicaid, they're just going to stop helping you out. And that's a huge deal. If you don't know, that's a huge deal for a hospital, man. Really big deal. So a lot of those larger hospitals are definitely striving to meet those requirements based on that, because they need whatever help they can get for themselves and their patients, right? Then we have adoption of universal precautions. So medical asepsis, so typical, we're going to go through these in a moment, and then surgical asepsis, and then the importance of the infection control officer. Medical asepsis is just talking about hand washing, disinfection, sanitization, and patient isolation as needed. If you have somebody who has developed MRSA, isolating them, you don't want them in the same room as somebody else, that sort of stuff. And here we're also talking about cleaning the linens on the bed and surfaces that patients could have come into contact with that aren't necessarily considered medical equipment, right? That all counts as medical asepsis. Then we have surgical asepsis. Here we're trying to keep an actual sterile condition as much as possible. We're cutting somebody's body open. We don't want to introduce any bacteria if we can help it. So we're actually trying to maintain sterility. And if what I'm saying about sterility doesn't mean a whole lot to you, sterile just is referring to having absolutely no living organisms, including endospores. There's none of them. So we have to verify that. We'll talk about that more in chapter 11, because that has to deal with, microbial control. But yeah, so we want things sterile. Then we have the infection control officer. Any of these rules, if you ever work at a hospital and they say, be sure you take your gloves off when leaving the rooms, be sure you wash your hands when leaving the rooms, be sure you wear booties in these conditions, all of that stuff. Those rules are all made and controlled by the infection control officer, not the nurses, not the doctors, not anybody else. It's all infection control officer. They're the ones that have the say over that stuff. Now, yes, your charge nurse might come and tell you, hey, be sure you wash your hands if they see you didn't wash your hands. But the person that made the rule, infection control officer, they're going to track potential outbreaks in the hospital like MRSA or C. diff, identify breaches in asepsis, people who aren't washing their hands when they leave patient rooms, for example, and then training other healthcare workers in aseptic techniques so we can maintain these good practices as we get new workers. So that's what the infectious control agents actually do. Next, we have reproducibility rate and the case fatality rate. So the reproducibility rate is called R naught, okay? R sub 0 or R naught. I always call it R naught. Sometimes I actually spell it out like that, just so you know, if you see it like that. Okay, so this is the reproduction rate of the illness, the pathogen, basically. So if you're infected with COVID and you're walking around and doing your life and whatever, or even, I don't even know, living your normal life, let's say, how many people are you likely to infect during the course of your entire infection? That's what R naught is. So as it says here, how many susceptible people on average that one infected person will spread the infection to? They're sick with a thing. How many other people are they going to get sick with it during their whole infection? So this is assuming that people you are walking by and coughing at and whatever, that they have no immunity against the microbe. So no vaccination and no natural immunity from having the actual illness. Okay, so that's what it assumes there. So those numbers are based on that. Next, we have case fatality rate. This is the number of people who die from the disease divided by the number of people who are infected. This is the likelihood that you will die if you get the disease. Okay, that's what that number is. So if you have it, what's the likelihood you're going to die? This is based on people who received no treatment from the disease. Why do we look at it this way? Because of, like if we look at, assuming you haven't been exposed at all or have any vaccine or whatever, well, most people do maybe for whatever disease. And then people who have received no treatment. Well, most people are going to go to the doctor probably. Well, yes, but what about whenever COVID first started? How many people were vaccinated? How many people were immune from having been exposed? Zero. How many people probably went and sought treatment? Probably not as many as you might like to think, right? They probably stayed at home and tried to tough it out because most people had flu-like symptoms, but then their grandma got it and died from pneumonia back whenever symptoms were awful. So that's why we look at them from like the worst case scenario, right? All right, this can vary by geographic location, the circumstance of how the outbreak is happening, and whatever variant of the micro we're looking at. We've already known that about variants of COVID, you know, with Omicron and Delta, some of them are more infectious than others. All right, so this is just a table that's showing us some comparisons. This is the deadliness. I know it's kind of harder to see up there, so I'm gonna write, oh, my handwriting is just wonderful, isn't it? Deadliness of it. So of course we know rabies, if you start showing symptoms, 100% fatal. So same with HIV. If you get infected with HIV, you got it forever, and you will die if you don't receive treatment, because that's what this is looking at, right? If you don't get treatment. Other illnesses could fall into that category as well. Next below it, we have tuberculosis, Ebola, and plague. People do survive these diseases without treatment, okay? Then we have syphilis. 25 to 49% of people will die from having had syphilis if they're untreated. This is usually due to neurological issues that are associated with it. It takes a long time to get to that point, to the dementia and everything, but you absolutely can die from syphilis. Next, we have the 0 to 24%, and I don't like to point out that 24%, y'all. I already tell you 20% of people die from this disease when they contract it. That should terrify you. That's 1 in 5. So that's fallen into the bottom of this category, okay? All that other stuff above it is not, you know, it's way worse. So here we're talking about not all of them, okay? Some of them are 0%, but rhinovirus, probably not that serious. That's just the cold. It causes a common cold. It's one of the causes. Polio, kind of pretty serious, right? We have things like C. diff, depends on how you're getting treated. Smallpox, that was a terrible deal. That was like 30% mortality rate or something when it was at its worst. But they were able to bring it down. Chickenpox falls down in here because it's not very deadly, and all of these, whatever. So that is looking at the deadliness. Whenever we look over at the communicability, or are not, the likelihood that you're gonna spread it to other people, we have at the very top the extremely communicable diseases: measles, malaria, mumps, rotavirus, pertussis. You cannot get malaria from another person. Okay? You can only get it from mosquitoes, but if you live in an area that is endemic to malaria, I promise you that almost all the mosquitoes that you come into contact with could potentially infect you. It's pretty serious. That's why if you travel to an area where it is endemic, they will give you prophylactic drugs. Yes. Measles, highly contagious. So highly contagious. Again, this is looking at if nobody is vaccinated or anything like that, right? So we have to consider that. I'll come back to that one whenever we get to talking about the vaccine part of it, because that's important, right? Highly contagious. Chickenpox. So we put measles, malaria, mumps, rotavirus, pertussis above chickenpox, and it's a whole other category, right? So you know how contagious chickenpox is. I know how contagious it was because I got it, but it's very contagious. Down here, we have things like smallpox, tuberculosis, polio, rhinovirus. This is the common cold. Okay, if you think that isn't communicable, I hate to break it to you. Right, obviously pretty communicable, yeah? So not really as much TB, that's less so, but it is communicable. And then these ones down here, you're just less likely to come into contact with them, especially if you are just like living a traditional life without any risk factors added in. So rabies and syphilis and HIV and all these things are just less likely to come into contact with them in the now, right? Okay, COVID during the pandemic, like actually during it, it's almost impossible to figure out what the R naught is of something new that's like up and coming and everything like that. So in the beginning, we thought it was 2.025. So that's if you're sick, you're going to infect 2 to 5 people. But now we know Delta, the different variants have their own. So Delta is going to be 3 to 5. And then Omicron is at least 5. Okay, So it's very communicable, but we also happen to know that with the vaccines, symptoms are signs and symptoms, right? Way less severe. That's what's helping us out the most. You might still get it, but it's going to be less severe. So vaccines are going to play a major role in controlling these diseases. We're talking about communicability. We're talking about deadliness of diseases. We're really wanting to consider the fact that if we're talking about those, completely unseen disease, you don't have any immunity to it. So what happens if you add in immunity? So the whole goal is herd immunity, and that is its own thing. It happens in a community. It cannot happen if we have people who are not participating. So herd immunity, this is when a significant portion of a population becomes immune, and that limits spread of the disease. So if you go out into the real world nowadays, and most people are, we'll say most just for now, right? Most people are vaccinated for measles, mumps, rubella, right? And you're walking down the road, and here comes somebody that has the measles. What are the odds that they're going to infect somebody in that scenario? Like super low, right? For the most part, because most people are going to be vaccinated. So people who cannot get vaccines, they're tiny little newborn babies, they can't get vaccinated. We have elderly who, no matter how much that you vaccinate them, just aren't going to build immunity anymore because they're old. We have people who are actively pregnant who maybe they didn't get the vaccine yet, but now they can't because they're pregnant, and all sorts of things. If they're, have immune system problems or whatever else, they literally cannot get the vaccine, right? You need 95% minimum, 95% of the population to be vaccinated against measles to achieve herd immunity. So that you can imagine that's such a high number to achieve that when you look at people who can't get the vaccine, and then you look at people who are adding into that number with, anti-vax or other scenarios, whatever, then we have a problem. And that's where we get scenarios like the outbreak that we just had in Florida elementary school. Now, an outbreak of measles in a Florida elementary school, that means there's multiple children that were not vaccinated in order for there to be an outbreak, right? So that should be concerning. This is not a talk about anymore. If you want to talk about the safety of yourself getting vaccinated and the safety of your own family, and you would choose not to get vaccinated because this is such an unsafe thing. This is not talk about you and your life. This is talk about the entire population sometimes. Yeah, we're talking about getting rid of diseases that were disfiguring and deadly. We have literally eliminated smallpox from the planet thanks to vaccination. We are about to do the same thing with polio. We've already talked about how horrible that is. Imagine your life and imagine your child in an iron lung, So that's what we're talking about when we say that, we might not be achieving herd immunity. That's the concern. It starts with the highly communicable diseases like measles. You'll see that first, specifically because it is so communicable. Yeah, it doesn't need very many people to not be vaccinated in order to see it getting spread around. So anyways, you see here how this would work. Yeah, you have somebody who gets corona. These people in purple are either immunized or they have a natural immunity. They survived it and now they can't get it again. So now we see less people that are getting, here's somebody that got it, but most of the people are vaccinated, so you can't get to the people who, can't get the vaccine or it's not working for them or they're allergic or whatever it is, right? Whereas if you look at no immunity, now we've got so many people getting the disease, the orange guys, and people even dying from it, that even if you do have people surviving and getting natural immunity, it's not protecting anybody. So that's the issue with herd immunity. Make sense? All right. COVID vaccines, when they were developed, because it was new technology, the mRNA technology had been developed earlier, but they hadn't gone through any testing, hadn't completed testing yet. Whenever the disease, whenever COVID was coming out and it was beginning to be clear that we needed vaccines available immediately for our healthcare workers, our first line defenders, then they said most of phase 2 was complete. So they were fine with going ahead with phase 3. They did continue to finish phase 1 testing. They did go through all of it, but it just hadn't been finished before they started phase 3. People said that it hadn't been tested properly. Well, they jumped that gun just a little bit, but they got approval from the government to do so. And I don't know, I'm pretty thankful for that. I think a lot of people are. So I mean, we wouldn't be here now if it weren't for the vaccine, right? This wouldn't be happening right now. Yeah, so nearly all of the vaccine side, it says nearly 100%. I don't know. So can we just say 99? Side effects are seen during the first 6 weeks of vaccination with the COVID vaccines. Studying the effects of the vaccine after they were distributed, we can call that phase 4, basically trying to investigate what the likely issues could be after the vaccine. I had COVID arm when I donated. I mean, it was like one of the worst. It was almost my entire arm, like red and like just, I don't even know. That's whatever. I recovered, and the second shot, I had absolutely no side effects whatsoever. So I heard that's how it went. It's like if you're going to have a side effect, it was either the first shot or the second shot, but not both. So I don't know. Anyways, anybody can report adverse effects to the vaccine adverse events reporting system run by the government. Patients can do it. Parents can do it for their children. We also have healthcare providers that can do it if they see any sort of issue going on with their patients. So that allows you to report adverse events so they can keep track of that sort of stuff as part of the monitoring for their phase 4. Also, there is the Vaccine Injury Compensation Program. This is a government program where you can receive compensation if there is a proven cause that it was the vaccine that caused your problem, whatever it is, that costed you money or cost you permanent damage or something like that, right? Usually no court or attorney fees involved with that if you do pursue that. If you know anybody that needs to pursue that. All right, moving on to emerging and re-emerging. Emerging diseases are new discoveries, like when we first had SARS come out. And every time we have a new strain of flu, if we need a new vaccine for it, then that's a new strain. It might as well be a whole new disease. Your body's never seen it before. So that's an emerging disease as well. Sometimes it's not that fancy. Re-emerging diseases, these are diseases that affected the population and were uncommon, but now are becoming more prevalent as we travel, as there's habitat invasion, or there's development of drug resistance. And they get spread more than they usually would. Collaboration between the CDC and the WHO, the global disease detection, just trying to keep track of these, emerging diseases and stuff like that, and looking at morbidity and mortality rates for those. So this is moving on to the neglected parasitic infections. There are quite a few around the world, but the CDC has started recently a campaign against the neglected parasitic infections in the United States. These are all protozoa and helminths infections here in the United States. We see them mostly in poor neighborhoods and in populations where we have immigrants that may have these diseases where it's more prevalent there. So this will include, this isn't an exhaustive list, but this includes Chagas disease. This is the one that is from the kissing bug that bites you. Neurocysticercosis, this is caused by tapeworm. So normal, typical tapeworm, you know how tapeworms have the little pieces, right? We call them proglottids, has the eggs inside of it. And people used to like buy them in like a bottle and like take them to try to like have a tapeworm grow in them. And then, oh yes, they will eat up all my calories and I lose weight, right? This is not a new, concept. So if you ingest the tapeworm eggs without being like eating it as part of like a meal or something like that, right? I mean, like in flesh, right? Where you normally would be exposed to something like this. Then the eggs can, and the larvae can migrate into the brain and create cysts in the brain. And then if you were to rupture those cysts in the brain, the inflammatory reaction is going to be so severe that it could kill you like really quickly, like within minutes easily. So it's bad. They can't get it out of your brain if you get it. That's what this is. It's going into your brain. Pretty serious. This is in the United States. Toxocariasis. This is worms that travel through the tissues and can cause blindness. Why do you think they cause blindness, y'all? Them worms traveling into your eyes. Okay. No, thank you. Toxoplasmosis. This is the one with the kitty litter. Don't handle it if you're pregnant. And then trichomoniasis. We already mentioned this a little bit, especially in the eukaryote lab, right? We actually looked at Trichomonas vaginalis. It's a sexually transmitted protozoal infection and can cause premature birth in infected mothers. All right, next we have bioterrorism. This is the intentional or threatened use of microorganisms against basically population. So we did have bioterrorism actually happen on U.S. soil in 2001. There was a spread of anthrax within Congress even, and that happened here in the U.S. And then agroterrorism, this is talking about agriculture here. You might think like, okay, well, they're going to affect agriculture, but you probably should take a step back and consider how important agriculture is in the United States. If you don't understand it, it's at least on par with, oil and gas. Oil and gas brings in maybe a little bit more money at, face value, but yeah, we get tons of money out of livestock industry. So this is talking about destroying livestock, it's talking about destroying crops and that sort of stuff, and that would devastate the population. The United States would not do well at all. I mean, I would say it would be like a catastrophic event if somebody actually conducted true agroterrorism on the United States. So anyways, this could include something like this virus called Rift Valley fever virus. And this is what the virus that the girl, I started out working on that virus because that's how I learned how to do all my lab stuff. But it was a girl in my lab that worked on it, and she sat right by me. Anyways, it's a BSL-3 virus transmitted by mosquitoes, and it's found in the Rift Valley area in Africa, so near Kenya, okay? And it's prevalent in the mosquitoes there, and it is 100% abortive to calves, like calves, unborn calves. 100% abortive. It would also be, I had to bring it to you, 100% abortive to people. Okay? That should be very concerning. It can cause hemorrhagic fever in people who aren't pregnant, but you can survive that, right? But the devastation, right? So there's a concern there, of course, for people, but we're talking mostly also about the cattle industry. If that was brought over to the United States, we do have mosquitoes that can harbor that virus. They're capable of it. So, anytime that there is any sort of risk of that situation, anything that comes into the U.S. that needs to be inspected, live cattle and plants and things like that, goes through Plum Island. Plum Island is just basically like the income point on that side, at least on that side of the U.S., that east side. But anyways, they're going to check for things like Rift Valley fever virus. I had a student, one of my first semester, I think it was my second semester I taught here, who he was from Kenya and actually grew up around it and like saw like how devastating it was to like tribes in the area. And all that. So it's pretty serious. Agroterrorism, something to be afraid of. So what is the difference between point source epidemic and common source epidemic? And why is there a distinction? Why does it matter? Remember, point source, one source all at one time, right? One exposure, that's it. Common source, there is one source physically, but it's spread out over time, like people visiting the contaminated wells. Okay. Let's do 15. No, that's not the number. 25. Did I just close it? I did, didn't I? No, I didn't. This is going to be pretty much me clicking through a lot of slides, so just bear with me. Okay. First, the biogeochemical cycles. There's two cycles I want you to be aware of. Carbon and nitrogen. Don't care about the others, all right? Carbon cycle. This is basically going to be talking about CO2 in the atmosphere. We already know that that's there. That it's going to go into plants and through photosynthesis be incorporated into organic molecules, right? We eat the plants and organic carbon from those plants and organic carbon from those plants whenever we eat dead things, wherever you're getting it from. If you're alive, eating alive things, or if you're eating dead things, or if you're eating the plants, whatever, you're getting the organic carbon from originally, at least something like a plant, right? Some sort of autotroph. Anyways, then during that, because we're making energy, right, ATP, one of the byproducts of doing that in aerobic respiration would be carbon dioxide, right? That's no surprise that we exhale that as a result of aerobic respiration. So guess what? We exhaled carbon dioxide and it goes back into the plants. And then we eat the plants and we exhale the carbon dioxide, and so on and so forth. We also have other additions like volcanic normal combustion that happens normally in the world, as well as industrial combustion that can add in carbon dioxide as well. We know that there's a rise in carbon dioxide since 1960. This is the carbon dioxide per parts per million. I used to have this chart that would show like the temperature change over time of the whole planet, the average temperature of the planet. And I mean, it like just this little squiggle like around like during the months and over several years. And it just goes like super hotter, 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 hotter. And when you look at stuff like that, that is like actual just data that they plotted out. Nobody did anything weird to it. It really puts things into perspective about the effects that, you know, we're having on the climate, but not just us, right? There's other aspects to this too. Methane gas plays a part in the carbon cycle as well, but more importantly, it is a more potent greenhouse gas than carbon dioxide. So we're talking about the methane that's coming from ruminant animals like cows. That accounts for about 20% of global methane production. Talking about that, agriculture there. That should be significant. Okay, so interesting. Next is the nitrogen cycle. Here we're talking about nitrogen fixation. So let's just put in some nitrogen into more usable molecules. The ones I typically talk about are going to be ammonification, nitrification, and denitrification. I am not going to ask you to know the chemical equations or anything. Just know the terms, that these are things in the nitrogen cycle, okay? And how nitrogen is used in, life. So here's some rhizobia bacterium and legumes like beans. So the bacteria live in little nodules on the roots. And what they do is help convert nitrogen in the environment into a usable form for the plants. That's all you need to know about them. This is just going into detail about these ammonification, nitrification, and denitrification. Cool. I don't need you to know them, but that's there. Those are the terms. All right, soil microbiology. It's kind of cool. We have a whole lot going on in there. Look at all these organisms that can be in the soil. Isn't it neat? All right, mycorrhizae, don't know a whole lot about it, but it involves fungus and bacteria and all this stuff working together. There are bacteria that live deep in the earth. That was that slide. Okay. Water. Here's water. The dark blue is saline. Here, if you break down the fresh water here, we can break that down into ice caps and glaciers largely. And then we have groundwater and all sorts of other stuff there. The other category is where you will find the lakes, rivers, and swamps. Then you can actually divide it out, looking at it that way. We get a lot of our water from places like aquifers and rivers and like streams and springs, that sort of stuff. So aquifer is just a deep underground water, and it will resurface through springs and geysers and stuff like that. Care about these. The Dead Sea has a lot of salt in it. I bet you knew that already. There's a lot. Okay, this is just kind of cool. You don't need to know it, but I'm going to say it because it's cool. 10 million viruses per milliliter in ocean water. That is something, y'all. 10 million viruses per milliliter. A milliliter, in case you don't know, that is a cubic centimeter, a cc. So if you ever worked in a hospital, you know what cc is. That's the amount of water we're talking about. Or you can just think about a centimeter by a centimeter. So a centimeter is about the width of my pinky finger. So just make a cube out of that. That's how much water we're talking about that has 10 million viruses in it. So much new DNA that's been discovered and investigated that we don't care about because it's nothing to do with healthcare. All right. There are red tides that can affect the health of water. We look at the amount of plastic floating in the ocean. Obviously, this is a problem. Everybody knows that, right? So 88% of the ocean surface, 88% of the ocean surface is coated with microplastic particles. 88% of the ocean surface is covered with microplastic particles, y'all. There's not a whole lot we can do about that at this point. 88%. So this establishes reefs in some cases for microorganisms to grow and create new little like ecosystems that should not be there. It also blocks sunlight from reaching the ocean floor, and it creates this weird ecosystem, like I said, shouldn't be there, causes an imbalance, and can lead to health risk for invertebrates and fish. Plankton are just going to be involved in making oxygen. Typically, they're involved in photosynthesis. They're the number one, bringers of oxygen to the environment. So this includes photosynthetic algae and cyanobacteria, which are our phytoplankton, ones that are going to do the oxygen. Oxygen in the water is usually used up pretty quickly by plankton in the area. So we need to have some sort of ability as like, especially a lake, to deal with, that sort of stuff. So how much oxygen is in each level, I'm not going to ask you these terms, but if you're interested in them, oligotrophic, as nutrient deficient, and then eutrophic, excess nutrients. These are both bad. Okay. Both bad, like nutrients from fertilizer runoff and all of that. It shifts the ecosystem and makes it so that we don't have enough oxygen and stuff like that. And then we have death of fish and other organisms in lakes and ponds. All right, drinking water. Where does your drinking water come from? We said rivers, aquifers, and springs. Yeah, not very much of water on the earth is drinkable, right? So here's how we treat our water if you want to drink it. Okay, raw water, we collect it, go figure. Then we're going to coagulate. So we're going to have things settle out of that water, the solids, whatever it is. It could just be like sand and stuff like that, but we're going to separate it from the water. And then we're going to, as it coagulates and settles, we're going to remove the water from the settled material. Then we're going to filter that water through sand, gravel, charcoal, and things like that. And once that is filtered enough to remove enough material, then we will disinfect it with things like chlorine and UV light. And that will be distributed to your home or stored for later use, like water towers or whatever. So in quite a lot of the world, your drinking water is also going to be used as your dump for your solid or liquid waste. So your sewage, unfortunately. So what is sewage? It could be used wastewater. It's going to be containing chemicals, debris, and microorganisms associated with poop and stuff like that. And then there's a large amount of solid waste, dissolved organic matter, and toxic chemicals that can be associated with sewage as well. So what do we do with water? When you flush your poop down the toilet, what happens to it? Where does it go? This is where it goes. All of everybody's poop water goes through wastewater treatment plant and gets released back into the environment. And it has been doing so for before you were born. So yeah, so don't think that's some sort of weird thing that makes us nasty or whatever. It doesn't. We're cleaning the water so that we can return it back to the environment where it belongs. Don't worry, we're going to test it before we do that. So our primary stage, we're going to get rid of our large matter. That makes sense. Bottles and, large matter, poops. Then we have the secondary stage. This is biodegradation by bacteria and other organisms. And then we have settling of something called sludge. Okay, and I don't even know how to describe that. I mean, I guess it speaks for itself, right? So if we can't get rid of something like solid or degrade it to be, used, I guess, or removed safely, then the sludge is going to get broken down and it's going to settle. And we can actually remove as much water as we can. And this actually helps us to get as much water out of the waste as possible. So again, saving it for the environment. But the bacteria are going to decompose everything involved in there, include toilet paper, whatever is left there. And then we have the tertiary stage, stage 3. This is going to be filtration and chlorination, just like drinking water. Okay. So we use reclaimed water from wastewater treatment plants for watering golf courses and parks. And we also will release them into large bodies of water that just helps disperse it a little bit more into the environment. You can reuse the solid waste as fertilizer or applying it to land to make it more nitrogen rich. Whenever we discovered that COVID was in, wastewater, basically, that was a problem. Luckily, we had a plan in place to test for it and to eliminate it because of polio. So polio is spread, it's a fecal oral spread. So people would drink it from water. And so they had to learn how to eliminate it from water. And so that whole process was applied to COVID basically when it came out. So that was lucky that we had something like that available. So we can monitor, basically the RNA of the COVID virus to make sure that it's being eliminated. Oh my gosh, okay. We definitely want to be sure that we are releasing sanitary water into whatever communities, or whether it's for drinking or whatever it is. We want to be sure that it's sanitary and clean, right? There are things like Giardia, Cryptosporidium, Campylobacter, Salmonella, Shigella, Vibrio, which is cholera, Mycobacterium species, hepatitis A, and Norwalk. These can all be found in sewage. Okay, so that's concern. So we need to monitor for these things, but we can't necessarily just monitor for those things because there could be there are quite a lot more than that, right? That could cause illness. So we look for indicator bacteria, things that we know would be there, that would be present in the intestines of birds and other mammals, including people, that we will identify using routine tests for those organisms. If they pop up in certain amounts, then we assume that the bad guys do too, even if they're not popping up. Okay. So these are called coliform bacteria. Our indicator bacteria are called coliform bacteria. This includes E. coli, Citrobacter species, Klebsiella species, and Enterobacter. They're all just going to be found in like basically the colon. All right. So coliform, just like E. coli, like coli, that coli. Okay. So these are coliform assays. I've been using it since 1914 to look for our indicator bacteria. If the indicator bacteria are there, then we're going to assume pathogens could be there. The first method, the rapid method, just plating it onto a plate and looking for colonies. This isn't going to differentiate the coliforms from other species, though. Let's see. We also have the membrane filter method, taking water that's, it's all diluted. So there could be microbes there, but how do we find them if they're all diluted out in the water? We still want to find them. So we filter them and catch all of them in the filter, take the little filter membrane off, and you can put that onto an agar plate and then let that grow. And there's like a limit to how many colonies you can grow, colony forming units from those coliforms to consider them safe. Okay. Obviously, you need to have less for drinking water. You can have a little more for wastewater once it's treated. All right. This is just a picture of that. They take the little filter off and then they plate it on a thing and then count the colonies. All right. Effects of microbes in food. It's not all bad, right? Some of it's good. We have things like kombucha, pickled cabbage, maybe. I mean, this is like sauerkraut, right? I have not had kombucha. I have a can in my fridge to try. It was my first time trying it because I hear that it's like, sour, like vinegary, and I don't know how I feel about this. Is it? Okay. Because I drink a lot of wine, so maybe I can get used to that, right? Huh? So I've got one that's it's a strawberry lemonade, I think. So we'll see. I don't know. We'll see. I'll let you know how it goes. Okay, guys. All right. I would not be seen ever eating that, the Norwegian fermented fish. That's a bit strong for me. And then Limburger cheese is pretty strong also, notoriously so. But it gives your food those notorious flavors. So then we have in bread, this is going to be yeast, but it's a certain kind of yeast called Saccharomyces cerevisiae, not Candida. Okay, so Saccharomyces cerevisiae, they release certain forms of gas and volatile organic compounds that give bread that smell that it has. You can also have other organisms added in or replacing those yeast, like the Lactobacillus San Francisco. That's obviously sourdough bread related. Sourdough bread, it does not taste the same anywhere else in the world than it does in San Francisco. My family's from San Francisco, so that bread is completely different. It tastes so different there. If you ever go to San Francisco, get this freaking sourdough bread there. It tastes different. Okay, it's way better, way better. Anyways, and then we have beer and making beer. Basically, remember, we're going to be taking a 6-carbon sugar through fermentation. We're going to have our byproduct is going to be pyruvic acid, right? Because when you go fermentation, that's what you end with at the end of your glycolysis cycle. So what do you do with that pyruvic acid and the electrons that you got out of the process? Well, you just mix and match and basically end up with ethanol and carbon dioxide. So this is drinking alcohol. And then you have the fizzinesses from that also. Okay. They don't add that in separately. Anyways, there's all sorts of processes that you would go through. I am not going to go through the beer making processes because I don't know them, and I don't know them. If you're interested in them, please feel free to look them up and learn about them. This isn't for me, and I don't have the time really to worry about it right now, so I'm not going to. but it involves basically malting barley. So you sprout your barley, I guess. That converts starches, and so then there's going to be sugars available for mash. So that malted grain will be, I guess, warmed up, and sugar and stuff will be added to make it just work a little bit better. Heat it up, and then the enzymes won't work anymore from the microbes. Then we have wort, which is the clear liquid after we filter it. They will boil that to give it aroma and flavor with hops, and I guess, and then it gives it color as well as destroying any bacterial contaminants. So yeah, so wort is inoculated with then that yeast, Saccharomyces yeast, and then it will convert all that sugar in that wort and turn it into ethanol and carbon dioxide. Cool. I went through it and I almost understood it. All right. Next we have One Health, which is, it sounds cheesy, but I understand what it's about, which is basically saying all the health in the world is connected. So the healthiness of this ecosystem and the healthiness of that ecosystem, we're all tied together somehow. I don't think it means like butterfly effect. I just literally think it means like we should be concerned about the whole health of the whole planet as opposed to just like areas, right? Is that it? No. Okay. Pathogen evolution. I don't really know what to say about this. It's just changing over time. So let's talk about some of the diseases that it's going to bring up. Hantavirus. This is also really hantavirus, the virus I worked on, Rift Valley fever virus, all related. They're all like the same thing. Okay. They all cause hemorrhagic fevers. Hantavirus, pretty serious. It's kind of uncommon. You'll, like it says, 15% mortality rate just in that normal hemorrhagic fever stage. If it develops into hantavirus pulmonary system, now we're talking about a 50 to 70% mortality rate. That's nuts. It's their mice pee and poop, and it dries, and then you sweep it up, and then you get it. This is in the United States. This is in the Southwest. So we're talking about like Arizona and New Mexico area. All right, HIV, we already know this is like an encroachment on territory sort of thing. Simian immunodeficiency virus from chimpanzees. They're assuming that there was butchering of primates and exposure to blood there that got introduced into the African population and got spread around the world from then. Yes, it started out being recognized as GRID, which is gay-related immunodeficiency disease, until they learned that they were basically, it's not just that, right? Okay. Next is the mosquito-borne diseases, any of them, basically. We got more mosquitoes that are surviving because of the warmer climates as we have global warming, basically. That makes sense. So we have things like Zika, yellow fever, dengue, West Nile virus that are spreading and increasing in frequency. We are having a spread. It looks like there may be a spread of dengue fever into the United States. There's no vaccine or anything for that. It can cause a hemorrhagic fever. But yeah, right now it's just in Central America and South America. Yeah, whatever. We know about the Aedes aegypti. So that is the spread of the mosquitoes there, and the predicted continued spread as the temperature rises. So the number of days per year in which mosquitoes are active and able to transmit. Y'all, I was out at Overholser and at Arcadia. You heard me talking about this. There was mosquitoes out there, and it was like 50 degrees maybe out there, and there's mosquitoes. And now I live right by Overholser, like a street, like an actual neighborhood street away from Overholser. And I have so many mosquitoes in my backyard, and they are like the size of like freaking dimes. They are massive, and I'm not keen on this. I liked it better when I lived in Yukon and there was no mosquitoes. So I don't know how I feel about it. Okay. SARS, we think it came from bats. You know, you have your own opinions on where you think it came from. I already kind of said what I feel like. So these are some newly recognized diseases in recent decades. Yeah, so this is like similar to the brain eating amoebas. But some of these guys, like, I don't know these diseases, some of them. These, never heard of it until I lectured this semester. Like, this is like a new thing to me. The rest of them I'm pretty much, familiar with. Anyways, this is Middle East respiratory syndrome. It's on camels, weirdly enough, but it's pretty severe. Anyways, then we have re-emerging pathogens, things that existed that are coming up again because of whatever pathogen due to the weather changes and all that stuff. So chikungunya, this is in the United States also. That is also a hemorrhagic fever. This is what I used to study. I'm just going to point out because I used to study it. And I don't know, monkeypox. I know we know about outbreaks with that. Candida auris is coming up a lot. We used to think of Candida albicans as being like the yeast that everybody has. There is an outbreak essentially of Candida auris now that we're seeing quite a lot of. My mother-in-law has Candida auris infection. She's on Humira, so she has a lowered immune system, but she was diagnosed with Candida auris specifically. And so I don't know a whole lot about that. I don't really know about Candida albicans. And these are some of the most concerning emerging pathogens. Again, we have chikungunya, which is in the United States transmitted by mosquitoes. And then we have Crimean Congo, which is what I studied. And then Ebola and some of these other ones, Rift Valley, Candida auris. So explain the importance of monitoring indicator bacteria like the coliforms in the water sources. It's just like if we can see the indicator bacteria, then the pathogens might be there. So we're going to play for the first time in a long time a thing, this Kahoot. That's what it's called. So chapter 14 is the one I picked since it covers like the more complex info. Okay. Just real quick, we'll push through it and then get out of here and go to lab. Lab is short, by the way. Lab is going to be pretty quick. It's quiet. We need more sound than that, right? Got it. Okay. I'm going to go ahead and get started. Remember, you can join as we go. Who was Jon Snow? What did he do? So hot in here, dude. It might actually, I don't know. It might be hotter in here than the lab. All right, so we've got stop the cholera outbreak. That is correct. Good job, everybody. It's like easy to remember his name. All right, which term refers to the total number of existing cases with respect to the entire population? Total and entire. See if you remember which term this is. Okay, so this one is prevalence. I know it's just a matter of learning the terms. Incidence is going to be looking at the total number of new cases in a specific amount of time, like over a month or something, right? Morbidity rate is the same thing as that. Which type of outbreak occurs when pathogens come from a single source and all victims are exposed at the same time? One source, one time. I can't spell apparently. That's not anything new. All right. Yep, that's point. That's point source. Good job. All right, true or false, a pandemic is the spread of an epidemic across continents. All right, good job. Everybody got that one. Right. Which term refers to a disease that exhibits a steady frequency over a long time period in a particular place? That place sees that disease at that level for a long time. An old hat. All right, so this one is endemic. I don't know. This is terms. I have nothing to say here. Okay. What aspect of a test is determined by how many positives were incorrectly tested negative? So false negatives. Which test is affected or determined by the false negative? All right, so this one is sensitivity. We're so sensitive, we're going to pick up all of the positives. We don't want any of those negatives, as they would be false. Okay. What are Koch's postulates used for? Determine the causative agent. You guys got it. Perfect, perfect. Right. Two factors, exposure and disease, are collected at a single time with no reference to which might have occurred first. We're looking at two factors at once, without looking at which happened first. Excellent. This is cross-sectional because you're looking in at like just a piece of the thing. All right, next, number 9. Multi-select, there could be more than one here. Oh, we didn't even do this one. I keep forgetting that this is in here. Okay, well, make a guess, y'all. Hill's criteria. I cut it out because it felt really complicated. There was a lot going on with it. I don't remember what the answer was either. I always felt like it was the answers that sound exactly the same to me. I don't know, but whatever, it doesn't matter. Okay, that won't be on the test. Okay, so what term refers to how many susceptible people on average that one infected person will spread the infection to? Okay, so that is R naught. Good job, guys. Here's a little podium, and then you guys can take your picture and we'll head up to lab.